now that we've covered some of the acute brain disorders, we're going to talk about some other types of more chronic disorders, with the exception of this first slide is kind of, it's kind of in the acute category because we're going to be talking about a type of infection. So you probably have heard of the condition called meningitis. Meningitis, as the name implies, means inflammation of the meninges. And you'll remember the meninges is that three-layered protective covering that surrounds the brain and spinal cord. And specifically when we're looking at meningitis, we're looking at some sort of either usually bacterial or viral infection that's impacting specifically the leptomeninges. And don't feel bad if you don't know what the leptomeninges is. The leptomeninges is considered all the space occupied by the dura mater plus all the space occupied by the arachnoid mater. The two of them together, don't ask me why, are called the leptomeninges. They just are. That's their name. So when I get an infection in these two layers of the meninges, this is called meningitis. And again, as I said before, most typically it's bacterial or viral, the bacteria or a virus that causes it. What's interesting is depending on the age of the patient, there tends to be certain types of infectious agents that are more commonly seen at certain ages. So for example, if we look at college age students like all of you, the most common causative agent of meningitis for a college student would be Neisseria meningitidis. But if we were to look at, say, young children or older people, Streptococcus pneumoniae would be more likely to be the causative agent. Now, some of the, signs and symptoms of meningitis include things, of course, like having a really severe headache, severely stiff and sore neck, photophobia, that's a big one, which means simply the fear of light. So they very sensitive to light. People can be very irritable. They don't function cognitively like they should, and these are different symptoms that might suggest meningitis. But in order for a clinical practitioner to actually diagnose meningitis, they usually have to do what's called a lumbar puncture. And that's where they take some cerebrospinal fluid from down in the lumbar region of the spinal cord, and then they examine it. And what they're looking for is things like leukocytosis. If there's a really high white blood count, that's indicative of an infection in the cerebrospinal fluid. They'll oftentimes see higher than normal protein levels in the CSF and lower than normal glucose levels, particularly if this is a bacterial infection, because remember, bacteria love glucose. So it makes sense if you've got a bunch of bacteria in the meninges, they're going to be consuming glucose that might be present in the cerebrospinal fluid, and so glucose levels will be lower than normal. If protein levels are high, that's usually an indicator that those bacteria are producing certain types of metabolic waste products that are proteins that are accumulating in the cerebrospinal fluid. You might get a different profile if you were looking at a viral cause for the meningitis or a fungal cause of the meningitis. Typically, if it's a virally caused meningitis, those are usually self-limiting and they resolve on their own, but the bacterial ones are they're more challenging to treat and clear. If a meningitis infection is caused by bacteria, it's typically called acute pyogenic meningitis. Traditionally, although it's really not accurate, but traditionally, if the meningitis is caused by a virus, it's called aseptic meningitis, which of course it's not, but they were trying to get at the fact that there's no purulent exudate forming. If you look at this brain here, you can see there's a substantial amount of pus or purulent exudate forming that is formed at the base of the brain, indicative of that meningitis. Another type of disease I want to talk about just briefly that impacts the brain is a disease called Creutzfeldt-Jakob disease. And Creutzfeldt-Jakob disease, or CJD for short, makes it easier to say, is a disease that is very similar to mad cow disease, or what's called bovine spongiform encephalitis. In sheep, the same disease is called scrapies. And this is a disease that was not well understood for a long time. It was a mystery to researchers as to what the etiologic agent was for this particular disease. What they knew is that it induced rapid onset of dementia and rapid onset to death, and that it would create this sort of spongy looking appearance to the parenchymal tissue of the brain. And what they've eventually found out about the causative agent was this. In all this suite of diseases, CJD, mad cow disease, scabies, the underlying problem is something called a prion. And a prion is basically a form of a protein. It normally occurs in all our brains. We have prions present in our brain that are normal and healthy and needed. But that somehow, in some way, a prion in the brain gets altered, or an altered version of a prion gets consumed in one's diet, and it leads to a chain reaction. So here's our normal, healthy prions here. Here's our altered prion. And the theory goes that when these altered prions come in contact with the normal healthy prions, it causes a conformational change, and the normal healthy one becomes an altered one. And so we get this chain reaction where all these normal functional prions that are needed for proper brain functions suddenly get transformed into these altered forms that are non-functional. And over time, this leads to injury of the neurons. You start to see the accumulation of vacuoles in the cytoplasm, and the cells eventually die. You get dementia and eventually death of the patient. I'm going to talk about just a couple different types of central nervous system disorders that affect principally the brain. And then in a subsequent portion of this AV lecture, I'll talk about disorders that are more common to the vertebral column. So the first central nervous system disorder I want to talk about in this part of the lecture is multiple sclerosis. And multiple sclerosis is an autoimmune disease. And as you probably know, with most autoimmune diseases, it's a disease that tends to have its onset usually during childbearing years, 20 to 40 or so. MS is much more common in women than men. In fact, it's twice as likely to occur in a female than it is in a male. There's about 1 in 1,000 people in the U.S. that have it. And one of the unique features of multiple sclerosis is that it is episodic, meaning that people will go through periods of time where their symptoms worsen, and then it'll kind of remit, and the symptoms will lessen, but the overall progression is a downhill slide. In other words, you might have a period where your symptoms are really bad, and then good, and then bad, and then good, but always you're declining in overall function. You never, after each episode, the patient never fully regains the previous level of neuronal function they had prior to the episode. So this multiple sclerosis is a disease wherein we demyelinate the axons in the central nervous system. And you'll remember that central nervous system axons are surrounded by oligodendrocytes. That's the type of cell that creates the myelin sheath around those axons, and that those myelin sheaths are there to help promote speed of transmission of action potentials. So when we lose the axons due to an autoimmune response, this ultimately impacts the ability of the central nervous system to communicate between neurons. So how this happens is another one of those hypotheses that's up for debate. There are a couple ideas as to how multiple sclerosis might progress. I've given you a picture here that shows you a very popular theory, which is that some sort of genetic predisposition will predispose a person to have a inappropriate immune response when they are infected with a particular infectious agent. And for example, there's been a lot of speculation that perhaps it's the Epstein-Barr virus that might be the triggering environmental cue that leads to the development of MS. So some sort of infectious agent, possibly Epstein-Barr, infects the individual, and if they are genetically predisposed to have an inappropriate immune response, they'll have antigen-specific T cells that will cross-react. Those T cells will recognize the infectious agent, and there's something about that infectious agent that is very similar. Its antigen is very similar in shape to antigenic proteins that are on the surface of oligodendrocytes. And so what happens is the person has an infection. Here's my infectious agent here. And we get activation of macrophages. We get activation of T cells. We get activation of B cells. And then these activated T and B cells cross the blood-brain barrier, enter into the brain, and there they cross-react or erroneously identify the oligodendrocytes as a foreign agent, and they begin to initiate an inflammatory response. Macrophages are activated. They begin to secrete all the different kinds of damaging and destructive enzymes that we've talked about previously in class. And the end result is that we get this degradation of the of all of these myelin sheaths. And those myelin sheaths eventually get replaced with scar-like material that are called plaques. And this, too, impedes the ability of those axons to transmit action potentials down their length. So symptoms that you commonly see of somebody with multiple sclerosis include they oftentimes have vision problems with their eye. If, for example, their optic nerve is impacted, they'll have various motor issues. They have that steady decline in function that I talked about before. They're never quite recovered to full health. Unilateral vision loss, I said that. They can have things like sensory impairment along with a motor impairment, loss of bladder control, spastic function. And again, remember, they'll go through these episodes where they'll get a little better, but then they'll have another episode and things will get worse. So this is a progressive declining disease, and it's not uncommon for people to die from complications from this disease. This picture down here shows you a cross-section of a brain from a patient who died of multiple sclerosis. And what I want you to look here is right around the third ventricle here, right in this area. I hope you can see that there's a very reddish, blotchy area. If that were a normal, healthy brain, that should all be myelinated axons. It should be white, but obviously it's not. And the reason it is not is because this demyelination has occurred in that area, so it's lost its whitish hue. Okay. The next thing I want to talk about is dementia. And, I'm sorry. There's only one, there are several different types of dementia, but I'm only going to focus on one type of dementia, and that is dementia related to the development of Alzheimer's disease. Now, of all the dementias, Alzheimer's is the most common form of dementia. And by definition, what dementia means really is a person remains conscious, they're able to function, but they have severe mental impairment, and that mental impairment usually involves memory, recollection, being able to recall things, identify people, etc. So even though the person's conscious, they're really not able to process mentally very clearly, and as a result, it's very difficult for them to be functional in society. So with Alzheimer's disease, there are a number of different ideas about what causes it, and I'm going to talk to you about the current leading hypothesis. There are alternatives, and I also want to explain to you very quickly that we know for a fact only 10% of Alzheimer's patients actually have gotten the disease because of genetics, because they inherited some altered gene from their family members, from their parents, and that altered gene is called the ApoE4 gene. Not that you need to know that, but it's a altered version of a gene that helps in the production of a couple different things I'm going to talk about right now, and those include something called beta amyloid plaques and neurofibrillary tangles. Now, the difference between the two of these, they're both types of proteins that develop in people as they age and are highly prevalent in people with Alzheimer's disease and can be identified at autopsy though not so much before that point. They're both protein type molecules. The beta amyloid plaques are found externally to neurons. And you can see down in this picture here, if I put a circle around it, all of this kind of tangled-like material around this central neuron, all of that is beta amyloid plaque. So that's extracellular. Inside of cells, we get a very similar type of formation of protein macromolecules called neurofibrillary tangles. Now, you can imagine that for both these externally occurring buildup of proteins and these internally occurring abnormal buildup of proteins impact the ability of axons to communicate with each other and to function properly. And so, of course, this is going to impair brain function. And indeed, the first kinds of brain function that do get impaired during Alzheimer's disease development is higher order brain function. So the person can still remember how to tie their shoes, for example, or make breakfast in the morning. But if you ask them a more complex problem like, can you tell me the streets that you take on your drive to work, where do you turn, et cetera, that might be more difficult for them. All of those frontal lobe skills, those executive skills, are the first to become impaired with the development of Alzheimer's disease. And this is considered primarily a disease of age. Now, and what that means is that Alzheimer's disease is more prevalent, and you're at greater risk of developing it the older that you get. The genetic versions of Alzheimer's can have a much earlier onset because of a genetic abnormality, but for those that are not genetically induced, which is 90% of Alzheimer's cases, it tends to appear with age. Now, I want to look at for just a second about the hypothesis of how it is that we get these things called beta amyloid plaques. And the way this works is on the surface of all neurons, there is something called the APP protein, and that's what you're seeing here. And APP stands for amyloid precursor protein. And that's a transmembrane protein. And while its function is not clearly understood, it's believed that perhaps this APP helps in the formation of synapses between neurons, and it might also help with neuronal plasticity, in other words, the ability of your brain to learn new things and to adapt and change. So when beta amyloid plaques are broken down, and remember all macromolecules are broken down because over time all macromolecules degrade and age and they get recycled or broken down and they get replaced with new versions of the same protein. In a normal, healthy individual who is not experiencing Alzheimer's disease, the breakdown of an old or degenerated APP involves the use of certain types of enzymes called alpha secretase. And when I use alpha secretase, and I'm sorry this picture is a little small, but if you look on the PowerPoint that I posted for everybody, you'll be able to see it a little clearer. Alpha secretase has the ability to cut this amyloid precursor protein right there at that black line I just drew. And as a result of that cutting, and with the assistance of another enzyme called gamma secretase, which cuts down here right at this line, we get the production of nice soluble fragments that stay inside the cell or are external to the cell. And because they're soluble, they are easily dealt with, removed, recycled, etc., and they don't cause any problems for the person. That's for a healthy person. An individual who has Alzheimer's disease has a altered ability to break down old and decaying APPs. And instead of cutting them with the alpha secretase and the gamma secretase, they use beta secretase and gamma secretase. So we still get our cutting here at the gamma secretase, but we get the beta secretase cutting right here. And the problem with that is that now I don't have these nice soluble fragments that I previously had. Instead, I have beta amyloid. That's this guy here. And those beta amyloids begin to accumulate externally to the cell, and the insoluble portion from the other cutting gets internalized, and we get the formation of these neurofibrillary tangles. Now, that's one hypothesis. It's a leading hypothesis right now to describe how Alzheimer's occurs. There are many others out there to describe what a series of events happen. And there's debate that goes back and forth over which is more important. Is it the neurofibrillary tangles that are the principal cause of the dementia, or is it the beta amyloid plaques that's the principal cause? Does it have to be the two of them together? Does one precede the other? These are all questions researchers are still trying to piece together, and obviously they're important to understand if we're ever going to develop some sort of an effective treatment regimen for people with Alzheimer's disease. Finally, let's talk for a minute about Parkinson's disease. I'm not going to spend too long on this condition, but since it is one that is seen with some regularity among the elderly, I thought it would be worthwhile to just briefly discuss it. Deep in the brain, there is a region of the basal ganglia called the substantia nigra. Nigra means black, and the substantia nigra is the only part of the brain that actually has melanocytes producing melanin, and as a result, it has this kind of black banded coloration to it, and thus the name substantia nigra. The substantia nigra does a number of jobs for the body, but the one that it does that really becomes evident when the substantia nigra begins to malfunction is that it is really responsible for inhibiting unwanted muscular movement. Okay, the brain is constantly sending off signals to the body to stimulate muscle contraction. That's part of what helps us maintain that tonus you learned about in 201. And what prevents you from having your muscles be overstimulated and getting twitching sensations all the time, or twitching motions all the time, is the substantia nigra. And the substantia nigra secretes a lot of GABA, which is gamma-aminobutyric acid. That's one of the neurotransmitters you learned about. And GABA is, as we know, an inhibitory neurotransmitter. So the more GABA the substantia nigra secretes, the more it inhibits movement. It also utilizes something called dopamine to help in this inhibition of movements. So as a result of that, if the substantia nigra stops functioning, for example, the last disease I want to talk about briefly before we go on to vertebral column disorders is Parkinson's disease. And Parkinson's disease is a disease in which an individual develops a very jerky walking gait. They tend to have tremors, involuntary tremors, for example, in their hands. They move really slowly. They tend to be stiff. They have sleep abnormalities, depression, fatigue. But it's principally a motor central nervous system disorder. And it centers around the degradation or death of cells within a region of the brain called the substantia nigra. And the substantia nigra is a part of the midbrain. It's one of the many nuclei that are part of the basal nuclei. Used to be called the basal ganglia, but now they're called the basal nuclei. And these cells in the substantia nigra are dopaminergic, meaning that they manufacture and secrete dopamine. Now, if you were to look at a cross-section of a brain and looking towards the midbrain, you would see that right here there is a region of the midbrain that has a very dark line in it. That's the substantia nigra. Nigra means black, so this is black substance. It's the only part of the brain, by the way, that has melanin pigment present. And the substantia nigra, as I said, manufactures this dopamine. And what dopamine does is it inhibits unwanted muscular contraction. Now, the brain is constantly sending signals out to the body to constantly stimulate muscles at a low rate. This is called tonus, and it's what helps maintain muscle tone. But we don't want to overstimulate muscle cells. And so what the substantia nigra does is it modulates the amount of stimulation to muscles that the brain accomplishes. And so you can imagine that if The substantia nigra is using dopamine as its main neurotransmitter to inhibit muscle movement when it's not wanted. Then if the substantia nigra begins to die or degrade, this means we get a drop in the amount of dopamine secreted, and as a result, the person gets these different conditions like unwanted tremor, bradykinesia, which means slow movement. They get these this very jerky kind of gait where it's hard for them to get started walking. So it's really hard for them to get out of the gate, if you will. And these are all because of the lack of proper function of the substantia nigra. What I think about is interesting about this is that you have to have, a patient has to have somewhere between 50 to 80% of their substantia nigra dead before they will even really begin to show the signs of Parkinson's. And what that really speaks to, I think, is the great plasticity or the ability of the brain to adapt to changing circumstances and for other cells to take over where dying neurons have left off. What causes the death of these cells in the substantia nigra? There's who knows? There's a couple different hypotheses. Some think it may be a genetic predisposition. Others think there might be environmental components to this. Some people have suggested, for example, intake of caffeine diminishes your likelihood of getting Parkinson's. We know Parkinson's-like symptoms can develop, for example, in individuals who take certain types of cocaine and amphetamines. So there are a lot of different etiologic factors. Most of the time, the cases are idiopathic, meaning we really don't know what caused them. We just know the person now has Parkinson's.